So good afternoon, and thank you to everybody for being here. And especially want to thank the committee, the organizing committee of Braining, especially to Claudia for inviting me and for inviting me and offering me this opportunity for being here. I'm very happy, for it's an honor and a pleasure. And thank you to everybody for hearing me. So today I'm going to speak about behavioral tests, but behavioral olfactory tests. Well, my speech will be divided in three parts. First of all, I would like to explain why the olfaction is very important in neuroscience, and especially why it's very important when we use different types of drugs or substances that could alter or influence in the central nervous system. And then in the second and the third part, I'm going to speak about different olfactory tests. First of all, we are going to talk about easy or simple olfactory tests. And in the last part, I'm going to talk about the olfactometry. This is a more complicated test and more sophisticated test, but could offer us more stronger or more robust results for olfaction. Well, so let's go on. Why do we study the olfactory system? Let me start my presentation with these images. These images are different views of the brain of a mouse. We can see here, I'm going to set the, point, laser. Can you see the laser?
Yes, we can.
Okay, great. Well, I'm pointing to this part of the brain. This part is the olfactory bulb. The olfactory bulb is the first relation state of the brain of mammals, of vertebrates. And you can see here in this view that the olfactory bulbs are very huge in comparison with the rest of the brain in rodents. That is to say that the olfaction is the most important sense in these animals. I told you before. Yes, so mice, rodents in general, but I'm going to focus on mice, have as the main sense the olfaction. Well, and as we can see here, in a corneal section of the olfactory bulb stained with Niesel staining, this is a structure that has a layering, a cortical layering. This is a cortical structure like the cortex or even the hippocampus. Also, the olfactory bulb receives inputs and sends outputs. Input from the olfactory epithelium. The olfactory epithelium is the epithelium in the nose of the animals that takes the information of the odors of the environment. So the olfactory epitheliums send inputs to the olfactory bulb. Also, the olfactory bulbs send outputs to other parts of the brain, and also other parts of the brain sends inputs to the olfactory bulb. So the olfactory bulbs receives inputs from two regions and sends outputs. Okay, again, the olfactory bulb also receives new cells. As we can see here in the right part of the screen, there is a region in the lateral ventricles called the subventricular zone. In the subventricular zone, new neuroblasts are generated, and these neuroblasts migrate through the rostral migratory stream to the olfactory bulb. In the olfactory bulb, these neuroblasts migrate again radially and becomes different types of interneurons in the olfactory bulb. So the olfactory bulb is a neurogenic region. In sum, the olfaction is the main sense in rodents. The rodents are the main type of animals to research in preclinical research. The first relation station in the brain that's related with olfaction is the olfactory bulb. The olfactory bulb has a cortical layering. The olfactory bulb sends outputs, receives inputs, and is a neurogenic region. So the olfactory bulb is more or less a small brain into the brain, and it's very important and a good model to study in preclinical research. Okay. In addition, there are new techniques for delivering drugs, that is the intranasal delivery system. This is a very simple system for delivering substances or drugs that consists of having an animal, a mouse, softly anesthetized, and we have only to put a small or 2 or 3 small droplets of a substance in the nostril. As you can see here, the droplet enters into the nasal cavity and impregnates all the nasal cavity. So it's a very easy system to deliver substances or drugs. And as you can see and you can guess, this system is very useful for research in the respiratory system, but also for research in the central nervous system. In addition, we can deliver by this system, by intranasal administration, not only drugs, but also viruses or even cells. And which is the pathway by these cells go through the brain. So there are 2 or 3 different pathways, but the main one is through the olfactory nerve pathway and through the olfactory bulb. So this is normal that if we deliver different types of substances into the nostril of animals, it's normal that we have to study the olfaction, because these substances directly goes through the nasal epithelium to the olfactory epithelium and the olfactory bulb. So usually olfaction should be affected, not only impaired, affected, let's say affected by these intranasal delivery of substances. Okay, I have here some examples of our laboratory in which we have traced some cells labeled in green here in the 4 pictures of the screen in different layers of the olfactory bulb. And of course, this technique, the intranasal administration of substances, is not only used in laboratory, but also is implemented in clinical research. So, well, this is a technique that we use in the clinics right now. Well, so if we use this technique or others, we have some reasons to study olfaction. Okay, there are more reasons. Okay, I'm giving reason for the sessions for the olfaction because it's my field of research. Anyway, well, olfaction. This is very easy to study olfaction in humans. We can ask humans about different types of odorants, good odors like the odor of books, the odor of wine, the odor of deodorant. But we have bad odors too. Maybe the odor of vomit, the odor of sickening, the odor of the old shoes, the odor of sweat. Okay, you can ask a person, oh, how do you smell this thing? Oh, good, bad. Oh, it's very easy because we can speak together in different languages and we have artificial intelligence to translate it. But we cannot speak in the language of animals. And up to now, the artificial intelligence does not speak the language of animals. Up to now. Okay, so this is the reason why, in order to test olfaction in animals, we use different types of behavioral tests. So let's move to the second part of my speech. There are a lot of behavioral tests for testing olfaction. But first of all, I would like to present you 3 techniques that are very simple test, easy test to know how the animals can smell, how the animals have their olfactory capabilities. So let's move in these 3 types of techniques, and then let's move to the 4th type, that is the olfactometry. Well, the first easy olfactory test is the varied food test. This is a very, very, very simple test that consists of directly hide a chow pellet in the cage of mice under the shavings. And we are going to measure the time that a mouse takes to find the chowed pellet. That's all. That's all. Of course, we need that the animals have a food deprivation. Animals should be angry in order to have to like to look for the chow pellet to eat in. So 24 hours before test, we have to do this food deprivation. And of course, we have to measure the weight of our animals and check if they lost more than the 10% of their weight. If they lost more than this 10%, we should remove them from the experiment because of guidelines of bioethical committees. But in any case, this is very difficult because they are not starving. They have only hungry after 24 hours without food. Okay. After the measuring of this seeking for the chow pellet, we will have some information about olfaction. Of course, this test is very simple. This test doesn't give us information about other detection threshold, discrimination, and so on. This test only gives us an idea about if the animals have impairments in the olfaction. Here, I would like to show you this graph from a research of our laboratory, in which we have tested the olfaction of mice using this test. In blue, I'm presenting you the latency time, around 45 seconds, that wild-type control animals take to find the chow pellet. And here, around 100 seconds, we have the time, a delayed time in which another type of animals, mutants in the nose enzyme, neural nose enzyme, is another history, takes to find the chow pellet. So we can see here that these mutant animals have some olfactory impairment because they cannot find the chow pellet as well as the wild type animals. Okay, of course, we can introduce some variations in this test and we can hide the chow pellet not under the shavings, but in an arm of a maze. And we can measure the time that the animal takes to find the chow pellet, but also we can trace the tray of the animal in its pathway. Of course, this variation could allow us not only to test the olfactory impairments in animals, but also memory or olfactory memory. Okay, this is a modification of this simple test that could also work. The second one is the short-term olfactory memory. In this test, we are going to present the mouse one odorant, but different times, 2 times. Okay, this is also a very simple test. First of all, the animal through to a phase of familiarization. We are going to expose the animal to the solvent of the odorant. Usually the solvents are odorless substances. Usually they are substances like mineral oil. I repeat, odorless substances. That's only to get used the mouse to this substance. Maybe the substance is odorless for humans, but maybe it's not odorless for animals. So that's why we do this phase of familiarization. Okay, we can do this phase once or twice, 1 day or 2 days, during 5 or 10 minutes. But anyway, 24 hours before the first exposure to this solvent, that is to say, 2 days after this familiarization, we start the test. So we are going to exposure the animal a plate with an odorant, okay, during 5 minutes. And in this 5 minutes lapse, we are going to measure the time that the animal spends exploring this odorant. Well, then we are going to remove the odorant, we are going to remove the plate, and we are going to give the animal a break of 15 or 30 minutes. It depends on the variation of the task. Anyway, this is a short time. After that, we are going to exposure the animal again to the same odorant during 5 minutes, and we are going to measure the time that the animal is going to explore this odorant. As you can guess, control animals should explore less time the odorant the second time that we expose it. Why? Because animals, because rodents, mice are animals that are very curious, and um, if they explore a second time the same thing, okay, for them is somehow boring. In any case, I'm going to present you some results of another research of our group. Here we have used this test to measure the exploration time in these short memory tests in two groups, a group of wild type animals in black and a group of anosmine 1 animals that have some impairments in olfaction and things related to olfaction, like olfactory memory, something like that. Okay, we cannot find statistical differences in the first exposure to the odorant, in this case menthol. Okay, that's not relevant. But after 30 minutes, we found differences between the two experimental groups. As I told you before, control animals spend less time exploring the menthol, but anosmine 1 mice spend more time exploring the menthol the second time. So we could detect here some impairments in olfaction or in olfactory memory. Well, let's move to the third and last easy olfactory test. This is an easy discrimination test. In this case, we are going to expose the animal two types of odorants, two different odorants, but in a subsequent manner, in a sequentially manner. So first of all, we are going to make a familiarization period, exposing the animal to two plates with the same odorant, during 5-10 minutes. It depends on the proof, 1 or 2 days. Okay, this is a familiarization period. The animal is going to become familiarized to this odorant in two plates. I repeat, these two plates have the same odorant. Well, 24 hours before this familiarization time, we're going to change one of the odorant, and we are going to put another or different odorant. And now we are going to measure the time that the animal explores both odorants, the known one and the new one, in a 5-10 minutes lapse. Normally, normal mice, control mice, should explore more the new odorant. As I told you before, mice are very curious animals, and of course, if everything is okay, they are going to explore much more the new odorant, the new thing that is in its environment. Of course, we can do further tests. We can change again the odorant, the first one, the second one. We can make changes in concentration. We can mix the odorant and so on, whatever you can imagine to measure the olfactory capability of these animals. As you can guess, this test is somehow a little bit more complicated, and it's why we can test more things. We can introduce more variants in this test. Well, when we use two odorants is very, very, very important when possible, using enantiomers. What are enantiomers? Enantiomers are molecules that are specular images. They are more or less the same molecule, but not exactly the same. An example are my hands. My hands are more or less the same, but not exactly the same. They are specular images. So enantiomers are molecules that have the same molecular weight, okay, have the same volatility. This is very important because we are going to test the odors of two molecules with the same volatility, but they have different odor. Okay, the odor is close, more or less similar, but different, definitely. Here we have two examples of enantiomers. For instance, the carbon plus and the carbon minus. They smell like menthol, something like that, like, I don't know, like toothpaste, two types of toothpaste, for instance. And we have here the second example of limonene plus and limonene minus, that they smells like lemon or orange or something like that. So like citric substances. So the odor is close, is similar, but not the same. Do you understand? Well, but this is very important when possible. Some implementation of this test could be the following one. For instance, an enhancement that is using a cage with two floors. In the first floor, we are going to put the two plates with the odorants. And then in the second floor, we are going to put two grids, two filters, in order to avoid that the animal touch the substances, to avoid that the animal impregnates their paws or their snout with the substances, mix the substances, so on. This is only an enhancement, but the basis of the proof is exactly the same. We can introduce a variation of a maze, a three-armed maze, in which we are going to measure the time that the animal spends in any of the two test arms, the arm with the known odorant and the arm with the novel odorant. I give you here two examples of good olfactory memory or discrimination when the animal passes more time in the arm of the new odorant, and example of a poor olfactory discrimination or memory when the animal passed the same time in any of the two or the three arms of the maze. Other variation is to use more odorants, not only two, but three or four. And of course, we can do some kind of automation to this test. That's just putting an infrared or a laser beam in the entrance of each hole up to the substances with odorant, just to measure exactly when the animal introduced its snout to sniff the known or the new or whatever odorant. Okay? Well, up to now here I have presented you three simple small tests. And now I'm going to present you the olfactometer. The olfactometer is a device to measure the olfaction in mice. Well, this device comprises a protocol, a more sophisticated and more complicated protocol. Also, the device is much more complicated. And the animals have to learn how to use this machine. So it implicates a period of some days of learning. But in any case, this time, this efforts, I think that perfectly supports the results of this device, because the results for measuring the olfactory capabilities in animals using the olfactometers are much robust, and also we can measure olfactory discrimination, olfactory detection thresholds, and so on. Let's see it. First of all, I would like to present you the matching the first olfactometer with which I have worked. This is an olfactometer from the Institute Pasteur, and I worked with this device 10 years ago, more or less. Okay, I learned with this device. As you can see here, there is a cage for the animal. Here, there are a lot of tubes, a lot of bottles with odorants, more tubes, and automatic stuff, blah, blah, blah. Okay. Another generation of olfactometer is this one that we have here in the University of Salamanca. As you can see, it's more or less the same, a complicated device with bottles for odorants, with tubes, more tubes, a cage for the animal, more tubes, more wires, and automatic stuff, blah, blah, blah. This device has been developed not only for our university, but also for members of the Institute Cajal and members of the Hospital for Paraplegics of Toledo. And finally, I'm going to present you the device that we have here in our research institute. And this is the last generation olfactometer. I have worked with it this morning with very satisfactory results. Jorge knows it. And okay, the device is more or less the same, but the bottles and tubes are inside this black box. And the animal is going to be placed here in this cage. Well, it's also automatized, but it's a much more compact device. We can store it, we can move it, displace it, solving the problems of storing and space in the animal facilities. And also it has some implementations of connection. It's based in the Internet of Things, and it can be, okay, it has a lot of automations. It can be easily controlled by a laptop or a tablet or even a mobile phone. With a mobile phone, with a smartphone, you can control not only one device, but also 2, 3, 4, whatever. And you can control it from the University of Poland, devices that are here in Salamanca. So it has a lot of implementation or automation. In any case, the functioning of olfactometers is the same. This is the general schema of olfactometers. First of all, we have here the source of air, an air compressor. That's all. The air is filtered in order to be purified, well, and pass through different tubes until this bottle, odor saturator bottles. Here I have represented 8 of them, but usually we use only 2. We're going to explain it afterwards. In any case, in these saturator bottles, we're going to pack different substances with odors or without odors. And the air is going to be loaded of these odorants and passing through more tubes, more tubes, more tubes, until finally it reaches this big tube, this chamber, this odor sampling port. And as you can see here, this odor sampling port is attached to the cage in which the animal is located. So the odor sampling port has an opening here by which the animal introduces its snout in order to sniff the air that comes through here, so through the different tubes and the odor saturation bottles. And as you can see here, opposite to the entrance, there is a small tube. This is a regular tube. Throughout this tube, the animal is going to receive or not water from this is the reward of the test. This is the reinforcement. So as you can guess, the animals that are going to do this task are thirsty, and the water is the reward for doing the task of the olfactometer. The paradigm of the functioning of this device is a go, no, go paradigm. That is to say, the animal has to decide to lick this tube to receive water or not, depending on the type of stimulus, positive or negative, with one or other odor that comes from the odor saturation bottles and these tubes. Let's see it. Well, I have removed the big part of the schema, and I depict here only the odor sampling port and the entrance and the entrance of today's odor sampling port. Okay, so when the animal explores, introduce its snout in the odor sampling port and breaks this infrared beam. When this infrared beam is broken, the system starts. This is an automated system controlled by the software. So when this system starts, the air comes through the odor sampling port. This air could be loaded by odorants or not, but in any case, this air could be a positive or a negative stimulus. Well, if the air has a positive stimulus, a given odorant, for instance, the animal has to decide to lick the water delivery tube to obtain the reward, a droplet of water. Well, if the stimulus is negative, a negative stimulus, another odorant, odorless, whatever, the animal has to decide to retire its snout and restore the infrared beam. Well, now we can have 4 different types of responses of the animals. The positive responses are the following. Heat. This is when the animal licks the water delivery tube to obtain the reward in the presence of a positive stimulus. Okay? When there is a positive stimulus, the animal has to lick the tube for obtaining the water. Well, the second positive response is the correct retreat. That's when the animal retires its head from the odor sampling port in the presence of a negative stimulus. So it's not going to have water, the animal retires its head. Correct retreat, positive response. Right? Let's move to the negative responses. Miss. A miss is when the animal retires its head from the odor sampling port in the presence of positive stimulus. It has missed the reward. And the second negative response is the false alarm. That's when the animal licks the water delivery tube in the presence of a negative stimulus. It's not going to have a reward because it's a negative response. Okay, these are the 4 types of responses of the olfactometer. Now, we can calculate the accuracy of animals doing this task. How? We are going to add the number of hits and the number of correct rejections and divide this number between the total number of trials. And we are going to multiply this number by 100 in order to obtain a percentage of accuracy. Usually, in each block of presentations, we present 20 trials, 20 presentations of odorants, 10 positive stimuli, and 10 negative stimuli. Okay, that's why the number of trials are 20, and we are going to summarize hits and correct reactions and divide between the total number of trials. Is it understood? Well, we can move back if you want. Well, as you can guess, for animals, it's somehow complicated know how this device works. So they need training sessions. And these training sessions are 2 types of training sessions. The first type is without odorants. In this first step, animals need to learn how does the olfactometer work directly. First of all, we need to deprivate the water of the animals in order to make them thirsty, in order to encourage them to participate in this task. Of course, we have to measure the weight of the animals and check if they reach 80 or 85% of their weight. Higher or less of weight should be avoided because of the guidelines of bioethical committees. So, and also we have to keep the animal stables around this weight, weighting them daily. And we have to support them some water each day, of course. That's why this deprivation is partial. Usually we support the animals with 1 milliliter of water each day, comprising both the water that they drink in the olfactometers and also a complementary water. Of course, this is around 1 milliliter. Some animals need a little bit more, some animals need a little bit less, but always keeping them around this weight to keeping them active and encouraged to participate in this task, to lick the water delivery to seek for water. Well, when the animals are stable, this is 1, 2, 3 days in this weight, they have to learn how to lick the water delivery tube. Well, the software directly gives the animals water when they touch with their tongue the water delivery tube in the very beginning of this task. So the animals learn that, okay, I'm going to explore this cage, I'm going to explore through this hole. Okay, this is the odor sampling port. They don't know that, but way. Okay, here there's a tube. I'm going to lick the tube. Oh, water. Well, they now know that in this tube they're going to have water. Well, after 20 licks of this tube, we are going to move to this part. We are going to increase the time that animals keep its snout inside the other sampling port. First of all, 300 milliseconds. When the animals has done 20 lickings of with this last time, we are going to pass to the next time of waiting, 600 milliseconds. After 20 lickings, we are going to pass to the next one, 900, then 1000, then 1200. Why? Because when odorants, when we will support an animal with odorants, as you can guess, the air with odorants needs some time to go through all the tubes, more or less 1 second. That's why we teach the animals to keep its snout a little bit more than 1 second into the other sampling port to, okay, wait a little bit, wait until to detect what's coming here and to decide what we're going to do, leak or no leak. Is it understood? Okay, so this increase of time is progressive. Maybe the first day of training we're going to start without last time, then 300 milliseconds and 600. That's all. Second day we can start with 300. Third day with 600. Okay, this is progressive, and this training session takes around 3-5 days, more or less. So when the animals know that they have to leak and they have to wait a little bit more than 1 second, we're going to pass to the second step of training sessions. This is training session with odorants. Now we are going to present odorants to mice, and animals are going to obtain reward only in the presence of a positive stimulus, not always, only in the presence of a positive stimulus. Well, usually the positive stimulus is an odorant. We have used the carbon plus, okay, and we have used a concentration that it could be easily detected by animals. We have used a dilution of 10 times in mineral oil. Well, as negative stimulus, we can use other odorant, but usually we use odorless, just the solvent of the first odorant, mineral oil. So we are going to present different blocks of presentations. In each block, we're going to present 20 stimuli, 20 presentation of stimuli, 10 positive stimuli and 10 negative stimuli randomly mixed. This is very important, okay? Randomly mixed. Okay, we are going to do 10 blocks of 20 presentations, and we are going to have a score for each of these 10 blocks in each day, okay? Well, animals are going to learn that they are not going to have water always. They are going to have water only in the presence of a positive stimulus, and progressively they are going to increase their accuracy in this device. And we can estimate that the animals have learned how to do the task when they reach an accuracy around 80-85%. Well, at the very beginning of this training session, the animals leak randomly the water delivery tube, and they obtain a score of approximately 50%. This is randomly, but progressively they increase their accuracy, and the score increases too, until, as I told you before, around 80-85% or even higher. There are animals that do the task perfectly. Well, these training sessions takes around 4 or 5 training days. And now the animals know how to use the olfactometer. And now we can start to play with odorants. Right now, it's okay. First of all, we can determine the odor detection threshold of animals. How? This is very easy. It's varying progressively each day the concentration of the positive stimulus. The positive stimulus, if you remember, is an odorant. In this example, the plus carbon. So the first day, we use a dilution of 10 times. Okay, and we will have 10 scores. We can do a mean of this 10 scores of 10 blocks of the first day. Well, the next day, we're going to present 10 blocks of 20 presentations, each one, but using as positive stimulus the same odorant, but more diluted, 10 times. Third day, we are going to do the same, but with a higher dilution, 1,000 times. Fourth day, the same, but with a higher dilution, 10,000 times. And finally, we can dilute it even more and 100,000 times. We can do further dilutions, but it's very, very, very difficult for animals to detect them. Of course, it depends on the odorant, but the plus carbon is more or less, this is the limit of detection. But in any case, we can determine the threshold of detection of different types of substances. And also, we can check the odor discrimination of animals. In this case, after this test, we're going to introduce a second odorant. In this case, it's very important that the second odorant is also an enantiomer, because we are going to mix this odorant, right? Well, so in the test for odor discrimination, we are going to use as positive stimulus the carbon plus, the same as before, and as negative stimulus, the other odorant, the other enantiomer, the minus carbon, okay? Well, of course, the concentration of both stimuli, of both odorants, should be a concentration that animals can easily detect. I don't know, maybe a dilution of 10 times or 100 times, more or less. Well, in any case, the first day, we're going to use as positive stimulus only carbon plus, and as a negative stimulus, only carbon minus, right? Next day, we are going to mix the odorants. And as positive stimulus, we are going to use 80% of carbon plus and 20% of carbon minus. And as a negative stimulus, 80% of carbon minus and 20% of carbon plus. Next day, we are going to use as positive stimulus 68 of carbon plus and 32 of carbon minus. And as a negative stimulus, 68 of carbon minus and 32 of carbon plus. And then last day, we are going to use as positive stimulus 56 of carbon plus and 44 carbon minus, and as a negative stimulus, 56 of carbon plus, minus, and 44 of carbon plus. As you can see here, the last day of testing, all stimuli are more or less the same. And remember that both enantiomers have a close similar odor. We can also do this task more complicated. I don't know, 5446, 5248. We can complicate the task progressively. But in any case, we can determine the threshold of odor discrimination. And for end, I'm going to present you some results of our group in which we have tested the odor detection threshold and odor discrimination threshold of wild-type animals, and also some mutants, PCD mice, that lost their mitral cells. These cells are projecting neurons of the olfactory bulb, and these animals have some impairments in olfaction. But it was the first time that these animals were checked in order to see how do they smell. So let's focus only in these graphs in the light blue lines and the red lines. We can see here the accuracy, the percentage of accuracy of wild-type animals in blue, and the accuracy of PCT animals in red. We can see here that wild-type animals have an accuracy around 100% and a dilution of carbon plus of 10 times and 100 times, around 90% and dilution of 1,000, and more or less the same accuracy and a dilution of 10,000. And of course, the accuracy drops a lot at the final dilution. And mutant animals have a lower accuracy in comparison with wild-type animals. Okay, concerning odor discrimination, we have here the different mixtures of odorants, and we can see here that wild-type animals have an accuracy perfect when the odorants are not mixed in the first mixture, maybe in the second mixture, or even in the more complicated mixture. Wild-type animals have an accuracy around 90%. They can differentiate perfectly these two mixtures. Maybe after that would be called complicated, but we have not checked it. PCD animals do it worse. Okay, but at the first mixtures, they can do it well, around 85%. They can discriminate odorants. Well, and just for curiosity, I'm going to moving back. In purple, I can show you the accuracy of some PCD animals that were subjected to cell therapy. And as we can see here, we can note a recovery in both parameters of odor detection and odor discrimination. But this is another history. That's all, friends. I would like to present you the group of the Institut Pasteur that helped me with the learning of olfactometers. This is the group of Pierre-Marie Lledó. As I told you before, Institut Pasteur, this is a group devoted to olfaction, a very good research group. And I would like to present our research group with a photo family, but we don't have the photo of the whole group, so I have done this collage. So I would like to give thanks especially to Jorge Valero for inviting me to this talk. This is a member of our group, and that's all, friends. Thank you very much for hearing me. Thank you very much for coming.